Hi, good afternoon. My name is Eva, so I'm from the Center for Brain Health in UBC under Dr. Utang Wen's lab. So I'm here to discuss our research on the pathogenic effect of the GABA alpha-1 gain of function in the early onset epilepsy and how we are characterizing this mutation to improve the patient's clinical outcome. So I'll skip over this page because the presenter before me have talked about this extensively. So our work flow is pretty much there's epileptic patient and they did a whole genome sequencing, and then they reach out to us to fully characterize them in vitro, and we also try to find ways to improve the clinical outcome. So to do this, we characterize them. We study the functional effects of the gain of function mutation on the receptor channel gating, kinetic, surface expression using in vitro expression system. We also screen approved drug to find negative modulator for the alpha-1 receptor, which can restore the gain of function mediated by the variant back to the wild type level. We are also doing generating alpha-1 gain of function mutation knock-in mice to probe the possible pathogenesis of the gain of function variant. And when we have the knock-in mice, we also use pharmaceutical intervention by applying the negative modulator to the knock-in mice during embryonic and early postnatal stages to reduce the early mortality. And then the last part is more we are currently doing to perform the electrophysiological, biochemical, and immunostaining experiment to examine the changes in primary neurons culture from the embryonic knock-in mice. But like the previous presenter said, it's really difficult to work with this knock-in mice, so this part is pretty slow. So during our characterization of a lot of the alpha-1 gain of function, loss of function variant, we kind of discovered that using the EC50 as the only indicator to characterize the alpha-1 gain of function is not very accurate in our hands. And we also find that the landscape of the variant functional response in the electrophysiological landscape is more complicated than we thought. Oops. Okay, so traditionally, gain of function is defined by EC50. So it is a measurement of ligand binding to the receptor. And like the previous presenter said, EC50 difference that's the delta log EC50, if it's larger than 0.2, it will be considered as a gain of function variant. And if it's larger than 0.7, it's considered as a high shift gain of function variant. And there should be, okay. So EC50 is closely related to the Hill equation. It's the inflection point of the Hill equation. And this equation on the mathematical level, it refers to the binding of the ligand to the macromolecule as a function of the ligand concentration. In other words, when the variant changing response is not only mediated by ligand binding, EC50 would not be able to capture the whole picture. So let me illustrate this with this example. This is the alpha-1 T257R variant. When you look at the amplitude of the current, these are the black is the wild type, and the red is the variant. You can see that the amplitude at all the GABA concentration from 0.1 to 1,000, the variant has a lower amplitude at all concentration. So this is graph against the wild type. But when you do the I over I maximum curve, which is the curve that is usually used to represent the EC50, you can see that this has a left shift curve. So it will be considered as a gain of function using the EC50 guideline. But in fact, you can see again, at all GABA concentration, the variant has a way lower amplitude than the wild type. So here are three more examples of the variant that we have characterized. So for all three of these variants, they all have a lower amplitude than the wild type at all GABA concentration again. As you can see, when we normalize it to the wild type maximum, the blue line, which is the variant, they are all lower than the wild type at all GABA concentration. But when we do the I over I maximum curve, the EC50 resulted from this curve are all different. So for R214H, there's a right shift, so this would be considered as a loss of function. And then for the F173L, it's almost identical as the wild type. So this one would be a new mutant. And then for T257R, it has a left shift. So this would be considered as a gain of function. And then after we kind of put all the data together, we realized that the landscape is even more complicated than what we think of just gain of function, loss of function. So all the previous example I have shown would belong to these two groups. We have divided all the variants we have characterized into 6 groups. So the right-hand side, they all have lower amplitude than the wild type at all GABA concentration. And then for the left-hand side, they all have higher response than the wild type at all GABA concentration, or similar at the maximum GABA concentration, but higher response at the lower concentration. So for this one, they would be considered as a gain of function, and for this one, they will be considered as loss of function for the functional response, but not the EC50, not necessarily for the EC50 way of classification. But then when we look at these two types, it's really complicated because they either have lower response than the wild type at lower GABA concentration, but then there's a shift to higher response at higher GABA concentration, or vice versa. Higher response at lower GABA concentration and then lower response at a higher GABA concentration. So just by looking at this, it's really difficult to say if they are gain of function or loss of function. It's all GABA concentration dependent. So since the function of GABA-A receptor is to mediate the inhibitory transmission in neurons, we think that the quantification of the functional change should be based on the inhibitory capacity. So we think using the area under the response curve would be another way that can describe the functional changes of the variants of the variants. So basically, if the area of the wild type is significantly larger than the area under the curve of the mutant, then it should be considered as a loss of function. And if the area of the mutant is larger than the area of the wild type, then it should be a gain of function. If we apply this method to the slide that I've shown 2 slides ago, you can see how these are the area. So you can see if it has a larger number, then it should be considered as a gain of function like this one, if you compare this to the wild type and this to the wild type. And then for the loss of function, if you compare this to this number for the wild type to the mutant. So it would be more clear cut. And then for these two difficult cases, as we thought it would be, the number would be way closer. So it would be dependent on just doing statistics if it's significantly different or not to be considered as a gain of function or a loss of function. Okay, so this slide just to say that ligand binding is only one of the many factors that can affect a receptor's function, like protein expression, channel gating, kinetic, etc. So to examine the gain of function variant, we have in more details. We also measure the channel gating and channel kinetic properties of the variant. And in general, we found that most of the gain of function variant, they change both the channel gating and the kinetic. This data are for just the F325L gain of function variant. You can see how the kinetic and channel gating change, and they are all GABA concentration dependent. We also found that most of the gain of function variant enhance tonic inhibition. Since alpha-1 is usually considered as a synaptic express subunit, it blurs the functional difference between synaptic and extrasynaptic receptors. So it makes the determination of gain of function or loss of function more difficult. So this table just summarizes some of the variants our lab personally study, and some are from online literature. So we did the calculation using the area under curve method to determine if they are loss of function or gain of function. And we found that most of the early onset epilepsy patients, they will be carrying the loss of function variant and using the method we have. You can see there are more loss of function than gain of function here. Lastly, I just want to report on an alpha-1 knock-in mice that our lab have been studying. So it's the F325L knock-in mice. So there are a lot of death. 70 out of 76 of the heterozygous homozygous mice we got died within 2 weeks. And then another 2 died 2 weeks later. And among the survivors, most of them are wild type, and only 4 of them were heterozygous. And out of the 3 females that survived, 2 of them are infertile. And this is just a video showing the mutation severely affects the normal brain function. The mouse have severe epilepsy, even a heterozygous. We try to use a negative GABA modulator, TCC, to enhance to make the gain of function phenotype back to the wild type. So in vitro, in HEK cells, TCC can restore the normal function of the gain of function mutant receptor. But when we apply it to the knock-in mice at early postnatal days, it showed that it will increase the spontaneous seizure activity of the mouse. So this is, we apply TCC, and this is 10 minutes after the application. You can see there is a higher current than the wild type. So this type of mouse have a significant shift in the EI balance to the excitation direction during development. This is the mini EPSC and this is the mini iPSC. You can see that it has an increased neuronal excitability and then decreased neuronal inhibition. So our preliminary data show that the gain of F325L knocking, it downregulate the GABAergic synapse and alter the GABRA1 and glutamatergic synapses ratio in the development. So this is a summary of the presentation. The receptor functional changes is caused by the mutation are more complicated, and EC50 alone might not be able to capture the whole picture. The most of the gain of function change the channel gating and kinetic. It also enhances the tonic inhibition. And then the knocking mice has a significant shift in the synaptic EI balance to the excitation direction. And then we think that the disruption of the EI balance caused by the compensatory changes during early developmental stages may be the basic pathogenesis of most gain of function variant. And using TCC, we showed that negative GABA-A modulator, which can restore the normal function in vitro, may not be usable for in vivo or patient study. I would like to thank my lab members, collaborator, and our funding agency. Thank you.
Okay, we have time for one question.
Yeah, so I guess the first thing is a comment rather than a question, and that is that the EC50 is not a measure of the ligand binding affinity. It's a measure of both ligand binding affinity and the gating efficacy. It's an empirical plot. And yes, if you change binding affinity, you'll change EC50, but it's not a measure of that. The second thing is to come to your characterization of the variants. And just a technical question. So I saw there some of the variants had increased tonic current. How did you handle variants with leak current? So did you set, for example, so if you pull off a patch, what sort of current did you use as like a threshold to continue measuring the dose-response curve of the cell?
So we use the wild type current.
But when you have a variant, yeah?
Yeah.
You patch it, yeah? Then you do the voltage clamp, you'll get a current.
Yeah.
A lot of us would sort of, if you had a very large current, we'd say that's a leaky patch and move on to the next cell. Is that what you did?
Yeah.
And about what current would you use as a threshold?
Yeah, sorry, I'm not too sure about that because I didn't do the EFIS part of that. I can ask the person that did and get back to you.
Okay, so the thing is that's actually really critical when you're measuring the maximum currents for gain of function variance, because if you have too much current, you may think that that's a leaky cell and move on to the next one. And what you invariably do then is you completely change your maximum current measurements, not because the variant has altered the maximum current, but because you're deliberately choosing the cells with lower maximum currents and lower surface expression because you think they're leaky. And when I see your data and I see concentration response curves that aren't going to zero and high levels of current, I concern myself that that is not being measured correctly. And I think that's a really important technical point which we've come across and has obscured the data. The second point is when we look at the dose response curves and you measured the area under the curve, I'm a little bit curious why you would do that analysis when on the y-axis, you've got a linear scale and on the x-axis, you've got a log scale. How did you account for the difference? Because that's, you don't want to be treating them the same when they're so obviously mathematically different. So why did you do an area under the curve? Why is it analysed as an area under the curve when you've got one side as a linear scale and one side as a log scale?
It's just that the area under the curve should be a good indication of the functional response of the receptor.
But why? I don't get it.
Maybe you should discuss it in a break.
Yep. Okay, yeah, so we will break for coffee and have 10 minutes.